Hello all, welcome. In this video we are going to see about chick development from Developmental Biology Unit 5 CSIR-NET Life Sciences portion through previous year CSIR-NET questions concept series. First question from CSIR-NET November 2020. Major structural characteristics of avian gastrulation is primitive streak, which becomes dorsal blastopore lip of amniotic embryo. Migration of these through the primitive streak is controlled by FGF8. What could happen if FGF8 protein, which repels migrating cells away from the streak, is overexpressed in the primitive streak? So in case of developmental biology, we have stages like morula, blastula, gastrula and norula. Morula, blastula, gastrula and norula. In gastrula, we see the formation of ectoderm, mesoderm and endoderm. For the formation of 3 germ layers, we need migration of cells, formation of dorsal ventral axis, formation of anterior posterior axis. These things will occur. There will be the formation of migration of archenteron. All these things are the flow of events that occur during gastrulation. So in case of chick, where the primitive streak is formed here and the cell starts migrating towards the periphery. If the migration is overexpressed, the migration is being controlled by FGF8. If this FGF8 is overexpressed than normal, all the cells will start migrate towards the periphery. This will be controlled by activation of WNT signal. This is the concept. Now coming back. What will happen if FGF protein, which repels migrating cells away from the streak, is overexpressed? WNT signaling will be activated and orientation of the primitive streak will change. B is the right answer. Next, from December 2019. Following statements are made about the events occurring during chick development. First, so we know about migration. Let's analyze the equations. Just now we have seen this. Inhibition of WNT planar cell polarity in the epiblast causes mesoderm and endoderm to form centrally instead peripherally. It's not inhibition, it is activation. So E is not right answer. C, B cannot be a right answer. This is the concept we have seen in the previous question. Okay. Hansen node of the chick embryo signifies a region at the anterior end of the primitive streak with the regional thickening of cells. Yes, D is correct. So what is Hansen node? You will see what is Hansen node. Consider this is a chick embryo. We have seen about primitive streaks and cell migrating. This is the anterior end. Anterior end, there will be a thickening. This end is called as Hansen node. D is the right answer. D is the wrong answer. By stage 13 of chick embryogenesis, little prior to the primitive streak formation of hypoblast is just complete. So consider the case during development. During development, the cells that form above is called as epiblast cells and the cells that forms below are called as hypoblast cells. The formation of cells below it, the colosicle here, posterior ridge here and all the formation will be complete by stage 13. This is the concept. C is the right answer. Now, development of primary hypoblast is mediated by the localized migration of the group of highly specified connected cluster of not 30 to 40 cells, it is 5 to 20 cells. They are called as islands. Fertilized chick undergo discoidal meroblastic cleavage. Okay. However, the cleavage does not extend to the yolk. Yes, correct. B is incorrect. E is incorrect. So, B is incorrect. E is incorrect. D is the right answer. Now, from December 2013. Hansen node is established as avian equivalent of amphibian dorsal bastopore lip. Following observation are presumed to be in support of the same. Okay. So, we know what is Hansen node. We have seen about amphibian development in previous videos. If you go back and watch that video, you can form an homology between Hansen node and sperm and mangrove organizer from amphibian embryo. So, it is a region whose cells can induce both the pattern when it is transplanted to the other location of gastrula. E is the right answer. It is equivalent in terms of tissue structure. This is irrelevant, incomplete statement. So, B cannot be an answer. Okay. It expresses the same marker genes as sperm and organizers. Correct. B is the right answer. Let's read the D option. The same microRNA can interfere with the formation of precaudal plate in both Hansen node and the sperm and there is some RNA involved in the expression, but not the same microRNA. If the same microRNA is produced, there will not be any differentiation between Hansen node and sperm and organizer. D can't be the right answer. B is the right answer. So, these are the 3 questions that came in CSIR NET GATE examinations right from 2020 to 2008. In my observation from 2020 to 2018. So, hope so you understood all the basic concepts of chick like Hansen node, Kolar sickle, posterior ridge, Highlands, hypoblast forming island cells. Hypoblast. Epiblast. Then about migration of cell, planar cell polarity, WNT signaling and FGF 8 signaling. These are the things, these are the concepts we get to know today in very short period of time. These are the benefits of going through the previous year questions wherein you can easily understand the concept. Hope so this concept is very clear with regard to the chick embryo development. If you think this video is very useful, please do like, share and subscribe to my channel and refer this video with your friends and fellow aspirants. Thank you for watching this video. Thank you.